Hello kids, welcome to part 2. It's a big part. It's going to be probably most of the period. Hopefully you've gotten these terms defined in your notes on page 5. The notes go in this part from page 5 all the way to halfway to page 12, the mid part of page 12, so up to slide 58. So please be ready for that. Okay, so now we're going to move on to slide 20. Now, we were talking about Mendel, right? Last time, Mendel Gregor, the Austrian monk. So basically he had 7 years of research, meticulous research, and the accumulation of pages and pages of data. We learned that many of it got lost, much of it got thrown out by the people who cleaned out his living quarters when he died, but there was obviously enough remaining. So from what was remaining, 3 very important laws of genetics were developed, okay? And they were the law of dominance, which we're going to learn about today. We learn about the next 2 laws, the law, we're also going to do the law of segregation and recombination today, and then we'll do the law of independent assortment next time, okay? So remember, laws describe a phenomena, but they don't explain the phenomena. The explanation comes from the theory. So that's pretty much the main difference. And theories don't turn into laws. That's a big misconception. Remember that. So let's start with the first one, the law of dominance. We talked a little bit about this last week when we did dominant alleles versus recessive alleles. So we're going to get a little bit deeper into that right now. Okay, so we are at the top of page 6, the new set of notes that you received today. So when an organism is a hybrid, big allele, little allele, that doesn't necessarily mean their size. That just refers to the capital letter and the lowercase letter, okay? An allele is a gene on a chromosome, which is DNA, right? Just trying to make sure you're on the same page, everybody. So a pair of contrasting traits, that's your alleles that are one maybe for short, one for tall, one for curly hair, one for straight hair, and so on and so forth, okay? It only shows the dominant trait, right? The other one remains hidden, right? That recessive one, the not expressed one, we call that dominant. We call those dominant and recessive. So these words, this is a little bit of a review, but more formally on the paper now, okay? So the allele that is always expressed is dominant, and the one that is not expressed, unless there are 2 of them, unless it's homozygous, remember our activity from last week, last Friday, those are recessive, the ones that are hidden. So here's an example. It's a very popular example with the Mendels pea plants, okay? A plant that always produces white flowers is crossed with a plant that always produces purple flowers. That's the homozygous dominant, pure purple, and the homozygous recessive, pure white. You take those and you mix them together, and you get what we saw last week, okay? Only purple flowers because the dominant gene, the dominant allele, dominates over the recessive allele. So only purple flowers are expressed. So in the F1 generation, the first generation after the parent generation, the pure generation, and the same thing can happen with people, we know this. Typically darker features like hair, eye color are dominant over lighter features. When offspring are born, they tend to have dark hair and eyes. This is not always the case, however, and we're going to discuss that. Oftentimes you will have something in the middle, okay? So what term is used to describe genetic makeup of parent plants? pure, homozygous. So a little bit of a review from last week's notes. The offspring has an allele for purple flowers from one parent and an allele for white flowers from the other parent. What term is used? It's a hybrid, right? If it's a mix of dominant and recessive alleles, it is a hybrid. And which trait is dominant here? White or purple? So there's a parent generation, the F1 generation. Obviously, the purple one. Okay, so that's it. That's the first law. Nice and clean. Now things get a little more complicated, okay? So this is the law of segregation and recombination. And that has to do with this, what we learned about last semester. I told you we were going to be relying on and applying some of that information from last semester to this semester, okay? So we all know what this is called. You don't know, these two sister chromatids are doing what? Crossing over, exactly, okay? And that happens during synapsis, in meiosis. And then they separate by disjunction. So it's the opposite of crossing over, okay? So each homologous chromosome carries one of the two alleles for every trait. So because of this law, segregation means to separate, right? Recombination means to put back together again. So because of these laws, you get a mix-up, a blending of traits, oftentimes. So in this way, alleles are separated and each gamete receives one copy of each allele. Remember, separation in anaphase, they're being pulled apart. First they're replicated, then they get pulled apart. Then they don't get replicated a second time in meiosis, and then these sister chromatids get pulled apart. And then recombination is fertilization, okay? During fertilization, they recombine from the parents to make that pair of alleles that expresses the trait. Could be dominant, could be recessive, could be hybrid of both, right? Okay, so you kind of get the idea here. That whole law of segregation and recombination. Segregation, recombination, okay? Stuff that we have learned about before. Applied to genetics. So here are some important terms, okay? A pure individual, homozygous, right? Pure means homozygous. For a genetic trait has two identical alleles, one from mom, one from dad. And that's called homozygous. Again, we touched upon these words last week. So we're just formally defining them now, okay? And the hybrid is called heterozygous. So here are some examples. I think I put these in your notes. On page 7, yes? So you've got just examples. You can see these in your notes. We've used these last week, okay? And then here are the pairs if they were located perhaps, for example, in those places on these chromosomes. So there are multitude, hundreds, thousands more of genes on one chromatid. And then the matching ones from the other parent are here, but they will be either recessive or dominant, okay? So the genetic makeup of an organism is called the genotype. We touched on that last week as well. The genotype are the genes. Gene, okay? And the outward appearance, like the appearance of someone's face, that F sound, phenotype. PH, F sound, right? Face phenotype. So that's what they look like on the outside. See, here's the genotype, here's the phenotype. genotype, phenotype, genotype, phenotype, genotype, phenotype, genotype, phenotype, okay? Get those 2 words down, because these are all the questions that are coming up are going to be based on all of this homozygous, heterozygous, genotype, phenotype, allele, all those vocabulary words, okay? But I think you guys got it by now. Here are some symbols that we're going to be using, okay? When setting up genetics problems, because the ones you got last week were very simple. They're going to get a little more complicated, maybe a lot more complicated. Letters are used, obviously, to represent the alleles. Again, this is all review from last week. For each trait, usually the first letter of the dominant phenotype, so I know you have blanks in your notes, but here, slide 29, so get those in. It's capitalized. The dominant one's capitalized, and the recessive allele is lowercase, okay? And here's a really good example with eye color. It's so easy. Just put, and when you're doing heterozygote, the capital always goes first, okay? Even though this is on top, don't write little b, big B. Don't do that. Not that it really would make a difference. It means the same thing, but we want to continue to be consistent. So when you see it on a test, hint, hint, you will know to write it the right way or understand what it's saying, okay? All right, so here's an example, another example. Lots of examples, lots of problems today. Slide 30. In terms of height in pea plants, the allele for T is tall, capital T, and the allele for short is little t, okay? And that is going to express, kind of gave him away there, okay? Big T, little t. And we're going to be completing this next chart on page 8 in your notes. So go to page 8. All right, so we've got this chart in our notes. The possible alleles, so if both are dominant tall, is that going to be pure or hybrid? It's going to be pure, right? Homozygous or heterozygous? They're the same. Homozygous. What type of genes? It's kind of given over there, okay? And phenotype, what is the physical look going to be? Short or tall? Tall. So I'm not doing these other ones for you. You are going to do those either on your own, now, but while you're moving on with the rest of the notes, or you're going to do it later, okay? Okay, so now we are on slide 32 and 33, gametogenesis. This is also a word we learned last semester. Gametogenesis is the creation of gametes, right? So a pure homozygous tall pea plant is represented this way. 100% of the gametes contain the tall allele, and 0% of the gametes contain the short allele. So the phenotype is all tall, and the genotype is all homozygous dominant. Big T, big T. And in a hybrid, heterogeneous pea plant is 50-50, right? They're still all going to be tall because of the dominant gene, but their genotype is going to be half short, half tall allele. So 50-50. All right, so let's apply this to human eye color, to the trait of human eye color. So we're going to use this information and all the knowledge we know about genetics so far, which is, believe it or not, more than what Mendel, Mendel, Gregor, knew, okay? So a young man has brown eyes. Maybe you have brown eyes and you're a young man. His mother has blue eyes. Raise your hand if your mother has blue eyes and you have brown eyes. Your biological mother. His father has brown eyes. Still? Is this anybody in this class? Assuming brown eyes are dominant and blue eyes are recessive. We don't have to assume. They are, okay? So you got dad, mom, and child. I put this in your notes. You can color the eyes if you want to, okay? So we're going to answer some questions about this scenario. What percent of the mother's eggs, ova, contain the allele for brown eye color? Remember, she is recessive for blue eyes. None, exactly. 0. If she had 1, she'd be brown-eyed, because it's dominant, right? What is the young man's phenotype for eye color, therefore? Well, not therefore, it was given to you. It's right there on your page. It's brown, right? What is the young man's genotype? Look at dad, look at mom, homozygous, dominant, heterozygous, homozygous recessive. He's going to be a mix of mom and dad. Big B, little b. And how do you know this? Because a mix of mom and dad, okay? Combined genes from both parents. Four, what percent of the young man's sperm will contain the allele for blue eye color, and what percent will contain the allele for brown eye color? If big B, little b is the boy, remember, okay? So it's going to be 50-50. Half the sperm are going to have the dominant allele, and half the sperm are going to have the recessive allele. What process will separate these alleles? Meiosis. We just went over it. Meiosis is the process where segregation or separation takes place. Okay? And where in the young man's body will this occur? The only place in the body where meiosis takes place. Everywhere else is mitosis. All right. So let's go to another scenario here, okay? We're going to use a Punnett square to solve some genetic problems. So in pea plants, the allele for tall plants is dominant to the allele for short. We know this already. Tall, short, okay? So the first step of every single Punnett square problem is to make a key. Here's the key. You're going to have what the alleles stand for and what the three different combinations would give you. And again, we did some practice with this last week, so this should be pretty easy stuff for you right now. And we are on page 9, okay? So let's go tall, short, sorry. Tall, short. Okay. You have to put those in there. But I mean, it's obvious, right? Tall, short, and then these two would result in a tall organism or offspring, and that one would be the only one resulting in a short offspring. So now let's go over the problem. Okay, so show the results of crossing a homozygous tall pea plant with a homozygous short pea plant. We've done this already. I'm just breaking it down for you, okay? So in your notes, you're going to draw what comes up here and here, okay? Or label what comes up there and there. So you've got the pure tall and the pure short, and these are going to be their choices. These are going to be the possible gametes, eggs and sperm, for the Punnett square, okay? that could possibly come together to make the offspring. So what do you do with this information? You construct a Punnett square. I put one on the right side of the page for you, in the middle of the page. And you're going to put the proper alleles. Okay? Parent number 1, parent number 2, and fill it in. Capital T, little t, capital T, little t, capital T, little t, capital T, little t. Make sure you're putting those on your paper on the right-hand side. And the 5th step is you take the information that the Punnett square gives you, and you could answer a multitude of questions from that information. Okay? So let's see what some of the information is. You're going to determine both the genotype and the phenotype of possible genotypes and phenotypes of the organisms, their probabilities, their percentages, and all of that. Okay? So what are the genotypes? How many homozygous, big, large, tall? 0, right? According to that Punnett square that you just made. But 100% of them are going to be heterozygous, and 0% are going to be homozygous recessive. Now the phenotype, remember phenotype is what it looks like on the outside, all of them are going to be tall. 100% tall and 0% are going to be short. It's pretty easy, right? I think this is pretty, once you get all that vocabulary out of the way, I think it's pretty easy. If you're paying attention. So, now we're going to cross 2 heterozygotes, okay? A mix of tall and short in their genetic makeup, and we're going to see what happens. So write this stuff down in your notes. This is at the bottom of problem 2, bottom of page 9. So you're going to, now we're going to have, in our Punnett square, we're going to take these possible gametes and put them on the top and the left. So you can start doing that now if you've already labeled your arrows and possible gametes, okay? So here's your Punnett square. You're going to put the possible gametes where they belong in order. 1 parent, 2nd parent. Doesn't matter whether it's the male or female. If you had questions about, is it the mom, is it the dad? You know, if there were questions pertaining to the parents, then you would have to be specific and know which one you're putting where and label this mother or father, okay? Or vice versa. So what are you going to have here? Homozygous capital. You're going to have heterozygous, and homozygous recessive, lowercase, right? So these are multiple questions. I could ask these in word form or in just like this, you know, kind of bullet form like this. So, or outline form like this. So what percentage is homozygous dominant? 25%, right? What percentage is heterozygous dominant? 50%, and what percent is homozygous recessive? 25%, okay? So what are the phenotypes? What percent are tall? 3 out of 4, right? So that's 75%. And therefore 25%, because all the percents have to equal 100, that's what this symbol means, out of 100. 25% are going to be short. So again, please know, this does not mean that if you and your mate have children, 3 of them are going to be tall and 1 of them is going to be short, and that's it. No. Again, it's like flipping a coin. It's the probability of every time you get a kid, or every time they have a pea plant, okay? You've got a 1 in 4 shot of the plant being short. 1 in 4 being short, and a 3 out of 4 being tall, okay? So when you're trying to say, well, my husband or my wife has blue eyes or brown eyes or curly hair or straight hair or tall or short, what are the percentages? What's the chance that our baby will have, and that's what this is. What are the chances that your baby will have these? You can't say that all of them are going to be a certain way. Okay, so remember, it's important to use the Punnett square. I was just talking about this, about the probability, okay? It's the expected results. Expected results. And the more, it's like flipping a coin, like I said last week. The more you do it with very high numbers, the more this probability, these percentages will be equal. Let me give you an example. Okay, so you're flipping a coin or you're rolling dice or whatever. I can flip 5 heads in a row on a coin. Easy. Or 5 tails, right? But if I did it 100 times, very high numbers, then the heads and tails numbers would balance to more of a 50-50. If I just did it 5 times and got heads for 5 times in a row, whoa, 100% of the time, I'm going to get heads. So if I have 5 babies and they're all blue-eyed, that means 100% of the time they're all going to be blue eyes. And you really can't say that because you haven't had a very high number of offspring, okay? That's what that means. So how do you determine the genotype of one individual person? So if a woman has brown eyes, that's her phenotype, right? By looking at the woman, can you determine if she's homozygous or heterozygous? Can you determine if she has big B, for her alleles from her mom and dad, or does she have big B, little b, one from mom, one from dad? You can't tell, can you? So the answer is no. So how can you determine this information? You would need the parents' traits. You would need to know what the parents were in order to determine which of these she was. So what's this first step all the time? Make a key, right? So we're going to talk about kitty cats. And this is what, so if you capital N is the allele for normal pigmentation, okay? Normal pigmentation would be like this, okay? Or just white, not albino. If, so dominant, right? Dominant recessive, so heterozygous, a hybrid. Also normal pigment. But if I get 2 recessive alleles, now I am homozygous recessive, and that means albino, an albino animal. Kitty cat here, with no pigment in the eyes, or very little pigment in the eyes, okay? Just because a cat's white doesn't mean it's albino. See the dark eyes? Okay. So look at the following diagram of the kitty cat family. Here's the mom. Here's the dad. Dad's what? Dad's albino, so it's going to be little n, right? Mom's going to be, whoa, it's not the right order, but that's okay. Okay. Oh, I see. Okay, so let's look at that then. Okay, so we know that we've got an albino cat here and an albino cat here. Okay, that's why we're starting with those first. Okay, so we're going backwards to figure out what the mom, because she's normal pigment, but is she big N or big N, little n, right? That's what we're trying to find out here. So we look at the kids, albino, we know these guys are normal pigment, but they're mixed. We know dad's albino, okay? So it's like a puzzle. They're mixed, so they're going to be heterozygous. And the only way that they can have a capital and none of them are homozygous, she must be mixed, just like these kids. So we use the kids actually here to figure out what the mom's genotype is. Because there's no way that she could have been two capital N's. Otherwise, all of the kids would be just like this with dad, right? Because they get X from mom and Y from dad, but there's no way, or X from dad. One of them would have to be capital N. Do you understand? If you don't, raise your hand and I will help you, or ask me later, okay? So which trait is dominant? Normal pigmentation or albinism? Normal, right? Which percent of the kittens are albino? What percentage of those kittens before were albino? 50%, right? What is the mother cat's phenotype on the outside? She was normal, but her genotype was heterozygous. Okay? Heterozygous. Both. And how did you know? How did you know that? Because she had to dominate an albino allele for her kittens to be albino. She couldn't be both capital N's. Otherwise, the dad, she gave birth to these kids. It would have to be impossible. What was the father's phenotype outside? What did it look like? Albino, right? The genotype would have to be therefore two recessive alleles. And how do you know? He has the recessive phenotype, so he must be little n, little n. We got one of these little cat ball rings things for my cat. She figured out how to reach in there with both paws and pop the ball out. And then she chases the ball around the room. It's really funny. And usually loses it somewhere, like under the couch or something. Doing good on this? Has recessive phenotype. Must be n, little n. All right. So what about cases where the hybrid offspring don't show dominant or recessive phenotypes, but something in between, like a mix, like this, like on the screen. Straight hair, curly hair, and then something in between, wavy. Red flowers, white flowers, and then something in between, pink, okay? This is called, we say rule breakers, but they're not breaking any rules. They're actually following the rules very well. We're gonna see how. This is called intermediate inheritance. In some cases, only one allele is partly dominant over the other. So it's kind of they both get expressed, okay? Halfway between. The offspring become halfway between the mom and the dad. So, incomplete dominance, that's what this is called. Incomplete means between. So snapdragon flowers, these are snapdragons. If you've ever seen those in someone's garden or in the outdoor part of Home Depot or Lowe's, okay? They have incomplete dominance. The red color is incomplete over the white color. And so R is for red and W is for white. So two capitals, two dominants will definitely be red, right? Definitely. Like that. And two whites will definitely be white, but the mix is in between is going to be a mix of the two. Pink. Like that. It's not an exact dominance. It's incomplete dominance. So I hope you're all following along. Slide 50. Told you this was going to be kind of fast. So we're on page 11. And we're getting into the first problem for this type of scenario, okay? So here's problem 1. Same thing as before. We're going to label it the same way with the arrows and the different gametes that the parents can produce, and then we're going to plug those into a Punnett square, okay? So what are the results of a cross between a red flowered plant and a white flowered snapdragon plant? And so the gametes are going to be capital R, capital R, capital W, capital W. And that's what you're going to put on the top and left of the Punnett square. That's on your sheet there, on the right-hand side, in the middle. And this happens during meiosis, segregate. The alleles come apart. They got these from their parents. So, plug them in. Doesn't matter which way you go. My example is this. So you're going to have all the same, RW, 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 and RW. They're all going to be heterozygous, incomplete dominance, and all of the offspring are going to be pink. All of them are going to be pink. Moving on to problem 2. Now, if you take 2 pink plants and cross them, so they're all going to be RW, and the gametes are going to be R and W for both, red and white for both. So now you can see what's going to happen. I think you're already in your mind at the point where you can visualize, if I cross these, I know what the result's going to be. I can picture it on the Punnett squares. But regardless, you have to fill it in your notes. So please put the arrows in those boxes under pink and pink on the bottom of page 11 and get those gametes into the Punnett square. It's kind of fast, I know, but you should be really good at this by now, all right? And what are we going to get? We're going to get capital R, capital R, capital R, capital W, capital R, capital W, and WW. So what are the genotypes? What percent are capital R's only? 25. What percent are heterozygotes? 50%. What percent are homozygous white? 25%. So what are the phenotypes? 25, 50, 25. So every offspring, the chances of them being red, 25%. The chances of the next offspring being pink, 50%. 25% red, 25% white. The next offspring, same thing. The chances of them being these colors are the same if you have the same parents with those alleles. All right, we are almost done. We have just a couple slides to go. Flipping the page to, I should hear pages turning, to page 12, top. Now we're going to talk about something called codominance. Not incomplete, but codominance. So in horses, the allele for red fur is codominant with white fur. It's not incomplete, it's called codominance. So the horses will have red and white or mixture. And in horses, it's called a roan. Anybody know anything about horses? I don't. I didn't grow up around horses. I think I've gone horseback riding twice, and both times were very stressful situations. Nothing crazy happened, but I would rather have been off the horse. Let's just put it that way, okay? And I know you have to be one with the horse to be a good rider, and I was never one with the horse. So, so we've got the R for the allele for red fur. So you're copying these down, okay? Capital R. Capital W for white fur. You've got homozygous, heterozygous, and homozygous, and those would be red, roan, or white. Just like red, pink, or white. You see, see how this is different than the pink? This is codominant, not incomplete. If this was incomplete, it would be one single color between these two. So it would be like a tan, I guess, in between brown and white. Be like a beige color, or there's a name for those kinds of horses, and I forget. Mrs. Keene would know right away. Mrs. Keene grew up with horses. I did not. And so you get both colors, pretty much like the black and white cat before. So red, roan, and white. Now it can happen in cows too. So you've got the brown and white, and then you've got I don't know if they call it roan in cattle. Anybody grew up near a farm or anything like that? I don't know what it's called. So here's problem number one. What are the results of a cross between a red horse and a roan horse? So this is going to be instantly in your mind. You know this is going to be RR and RW, right? And what are their possible gametes? Capital R, capital R, capital R, capital W. So plug these into the Punnett square at the right of the page, and you will get the results. So the genotypes are 50-50, right? 50-50, 0. No white babies. All red, all roan. This is how you've heard of horse breeding, you've heard of animal breeding, you've heard of the word thoroughbred, okay? You know about dog breeding, and you know that people do these things. This is what they know this. They know genetics. They're not geneticists. They're not working in a laboratory with cells and things like that. But they know that if you cross this with that, you can have this pedigree chart, and you could show that, and if you want certain traits in your animals, and even farmers in ancient times knew how to do this stuff. They were like, hey, I want a horse that, a Roman horse, whatever, that can travel long distances. Or I want a horse that can pull a plow that's strong. Let's breed them for the traits. They didn't know anything about this, but they did know about breeding, okay? And if you bred something for a certain trait, you could get those traits in the offspring, okay? So it's 50-50, 0. You should have already probably written that down already. Didn't need me to tell you that, okay? And that is our notes for today. That ends our notes. So we have one more part, or two more parts, actually, of notes left. Two more parts of notes. And they get a little more complicated as we go through them, all right? And we're going to have more practice, too. So tomorrow, we're actually going to do this next set of practice for these types of crosses, okay?